my doctor kept saying, I want you to make it to 34 weeks, and then 35, and then 36. Finally, at 38 weeks, I was starting to have some contractions. He was in a single practice and was going to be gone that weekend. And he delivered my first child, and I didn't want some stranger covering for him to deliver the twins. And so I said, can you induce them, please? And he said, okay, 38 weeks for twins. That's far enough along. Unbeknownst to me, my sister-in-law was 3 weeks late with her baby. And she had scheduled her induction for Monday. I was 2 weeks early with a scheduled induction for Friday. So my mother-in-law got 3 grandbabies in a weekend. And if we had scheduled it, we could have it all born on the same day. All right, but if a woman goes in, it's like a crochet hook. The doctor goes up in there and pokes a hole. So which of these layers, all right, would be poked or torn by that crochet hook to allow the water to leak out and induce labor? So definitely the amnion, because that's the membrane sac that surrounds the baby. Chorion levy, because that's this layer right here. What else? Decidua parietalis. Let's take a look at this picture. So the crochet hook is coming up here. Here's the chorion levy right here. And here's the amnion. What layer of decidua is this? Capsularis. All right, so it's going to go first through the decidua capsularis, then the chorion levy, then the amnion, and then the amniotic fluid. Chorion parietalis is along the lining of the uterine wall. And by access, if we came in, if we were doing a C-section, yeah, you would cut the chorion parietalis. But if you're coming up through the cervical canal, you're not going to penetrate that. Okay? So then we have there's some point I was going to make here. So this uterine cavity right here is just about obliterated, all right, at term. But it's still separate. So now the baby is born, the umbilical cord is cut, and this is actually ruptured. The baby has come out through there. And then we have the afterbirth. Right? So the afterbirth, as I indicated with you on Tuesday, is just like menses. With menses, we have separation of the stratum functionalis from the stratum basalis. Stratum basalis remains behind and regrows a new epithelial layer from the uterine glands. So after birth of the baby, the afterbirth is the placenta, stratum, the chorion villi and stratum basalis separate from the stratum basalis. Okay, the stratum parietalis separates from the stratum basalis. Remember, all of the decidua are just different areas of the stratum functionalis. So the placenta is the chorion villi, and what separates the true placenta is just fetal tissue. But when we look at the placenta outside the uterus, it's the chorion villi and the decidua basalis. That's going to separate from the stratum basalis. That's where it's confusing because we have two basalises. Okay, but the decidua basalis is really the decidual reaction of the stratum functionalis, and that leaves behind the stratum basalis, and all the decidual tissue separates like a regular menses, except there's just more tissue, and that's the delivery of the afterbirth. Okay. So we'll finish up this section here by looking at some of those structures. So this is the fetal surface. We're going to talk about the amniotic membrane. Placenta means cake. Some cultures actually eat this. Some animals eat it. It's very rich in protein. Okay. You can actually buy a shampoo called placenta. I would be grossed out every time I wash my hair with it. But when my mother worked as an OBGYN, they had a big 10-foot chest freezer, and they just threw all the placentas in there and sold them to a processing company that would process them for the proteins. The moms never got any of that money. So here's the amnion on the Navy side. So what we're looking at here is this surface right here. Okay, so the amnion forms a sac around the embryo, covers the placenta, and covers the umbilical cord. So when that's delivered, amnion. If you turn it over, this is the deep surface of the decidua basalis, where it's separated from the stratum basalis. Okay, so you're looking at maternal tissue when you turn the placenta over. So this was twins, 2 umbilical cords, same placenta, and these bumpy things are called cotyledons, but again, this is where, you don't have to know that word, but this is where the decidua basalis would have attached to the stratum basalis. That was left behind. Sometimes it doesn't completely separate, either the decidua parietalis and little pieces of the decidua can be left behind, and that makes it difficult for bleeding to stop. And the woman may continue to hemorrhage, and they'll have to take her back in and do a D&C. Make sure all of that decidua is removed. Same thing on that side. Showing the amnion on the surface of the placenta and the umbilical cord. Now this next picture was supposedly taken of a little girl's placenta, obviously a baby girl, born on Valentine's Day. I think it was just the daddy playing with Photoshop. Doesn't look quite, I don't know. I suppose that there could be heart-shaped placentas, but for all of that to happen on Valentine's Day, I'm a little incredulous. Okay, so maternal side, fetal side. So as it's delivered, the placenta is formed of both maternal, as the decidua basalis, and fetal, as the choriombili. Right? When we look at it in the uterus itself, when we're talking about what the placenta does, and placental tissue, we're talking most of the choriombili, the fetal villi. So if I ask questions about that, I need to distinguish which placenta, where the placenta is when we talk about it. Okay? Now, this fetal tissue is not, it's foreign to the mother. Why? Okay, half of the DNA on these cells did not come from her. They came from it. It's really a transplant. They came from the father's DNA. And so as all of this implantation is occurring, there is actual suppression of immune activity in the uterine tissue, so that her white blood cells and her lymphocytes don't attack it and prevent it from implanting. For some women, that does happen. They develop antibodies against proteins of their baby's father, and they reject and destroy anytime the embryonic tissue is trying to implant. All right? For other women, the suppression of that immune activity is systemic, and they develop cancers during or infections during the pregnancy period that they would not normally develop. It's like taking an immunosuppressive drug when you have a heart transplant or a kidney transplant. It suppresses your ability to attack anytime the body is destroyed specific molecules or pathogens that might attack the body. Normally that's reserved just for this decidual area, okay? But sometimes it can be system-wide. All right, let's stop there, take a break. And when we get back, what time we finish? Right. When we get back, we'll do about 30 minutes to do the embryo development. Okay, we're not going to go all the way to birth. It's going to do the first 3 or 4 weeks. So 20 after, okay, slightly more than a 10-minute break, 15-minute break.